Hi, my name is Farid, and I'm going to briefly talk about the research that I'm currently doing in the lab. I'm working on a protein that is called selenoprotein S or seleno S. Here in this figure, you can see the structure of this protein. It has 189 residues. It has a small segment that uh, attaches the protein to endoplasmic reticulum membrane. It has a coil cord region. It has 3D structure up to residue 122, and the rest of protein is intrinsically disordered. It doesn't have any specific structure. And in penultimate position, there is a selenocysteine residue. Um, The function and role of selenium in the cell is not completely understood, but what we know so far is that this protein is involved in managing ER stress. It is part of a process that is called endoplasmic reticulum associated degradation. This is a process that translocates misfolded proteins from ER lumen to cytosol to be degraded. It is also involved in managing oxidative stress, and it has relation to uh, inflammation. Actually, it has anti-inflammatory function. There is some evidence that this protein is connected with the replication machine of SARS-CoV-2. Uh, this figure shows this complex. NSP12 is the catalytic core, and NSP8 and NSP7 are cofactors. This is the complex that you can see here. Um, Actually, you can see the 3D structure of that. And there is some evidence that Selenos interacts with NSP7, which is this is small protein in magenta. Uh, to study that interaction, we used some experiments that are called pull down assays. We expressed NSP7 with an affinity tag. We also purified seleno S, and we mixed those two proteins together, and we applied that complex to affinity beads. And after washing, if uh, those two proteins interacting with each other, we can detect both of them in eluate. And actually, from these sets of experiments, we could find out that these two proteins, NSP7 and seleno S, are interacting. And now we know that the membrane part is involved in this interaction. However, this figure is only a model, it's hypothetical because we don't know which part of NSP7 is involved in this uh, interaction. And we also don't know if seleno S attaches this complex to another molecule or it attaches that to a membrane or not. Actually, these are the questions that we are going to answer in future.